Hello students, in today's class we are going to learn about temporary slide preparation for mitosis. So the aim of the experiment is to prepare temporary slides and study stages of mitosis in onion root tip cells. Requirement for the experiment includes one normal HCl, acetocarmine dye, burner, water, slides, cover slip, watch glass, dropper, needle and blotting paper. Coming to the principle of the experiment, after germination of the onion root tips, we add aceto-alcohol solution that is used for long-term preservation of the root tips so that they do not rot. HCl treatment is done to bring about softening of the roots by dissolving the cementing material, and acetocarmine dye is used for staining of the genetic material or the chromosome. Coming to the procedure, first we place the onions on a tile and with the help of a scalpel, carefully remove the dry roots of the onion. We then place the bulb in the beaker containing water to grow the root tips. It may take around 3 to 6 days for new roots to grow and appear. We trim around 2 to 3 centimeters of newly grown root tips and place it in a watch glass. With the help of a forceps, carefully transfer the root tips into the vial containing aceto-alcohol solution, which consists of glacial acetic acid and ethanol in the ratio of 1:3. We allow the root tips to stay in the mixture for preservation of the root tips. Further, we pick up one root and place it on a watch glass and add few drops of dilute HCl and few drops of acetocarmine dye. We flame it intermittently by passing it through a burner such that the stain does not dry out completely. We allow the HCl treatment for around 5 minutes. The excess stain can be carefully removed using a filter paper. We then trim off the tip portion of the root and transfer the tip to a slide, add a drop of glycerin and put a cover slip on the sample. We then squash the cells till it straightens out as a fine cell layer. We then observe the slide under the microscope and record the results and identify the stages. So this is a diagrammatic representation of how we keep the onions for formation of root tips. So we dip it partially in a beaker containing water and the beaker is kept in the sunlight. We can see after 1 or 2 days, formation of few roots will take place. Coming to the observation, so interphase is a stage that is prior to the actual mitotic stage and the cell appears to be inactive and in a resting stage, but it is metabolically more active. The nuclear membrane and the nucleolus are very distinct. The chromosomes are in the form of a chromatin network and the individual chromosome cannot be seen separately. The chromosomes appear double-stranded here. During early prophase, which is the first stage of the mitosis, we can see a clear nuclear membrane. It appears distinct. Nucleolus is also seen distinctly, but chromosomes become coiled and shorten and more distinct. During late prophase, we'll see that the nucleolus and the nuclear membrane have partially or completely disappeared. Each chromosome now begins to show chromatids, primary construction and centromere. The chromosome begin to move and gather near the equatorial plate. The chromosomes are condensed and thus shows up and thickens. The spindle fibers begin to appear. During metaphase, the nuclear membrane and the nucleolus are absent, having disappeared. The centromere of the chromosomes are arranged on the equatorial plate and each is attached to the spindle fiber. The spindle is made up of fibers only. The absence of centrioles indicate that it is a plant cell. The chromosomes of the metaphase are distinct, thus number, morphology, and chromosomes are studied at this stage. The anaphase stage is completed in a very small period of time. The centromere of each chromosome gets split into two. The chromosomes are also get divided into two chromatids. Each chromatid now have one centromere. The chromosomes become shorter and thicker, and the separated chromatids are now pulled towards the opposite poles due to contraction of the spindle fibers. Telophase stage represents the last stage of mitosis. The chromosomes are present at both poles of the parent cell. The chromosomes increase in length and become thread-like. All the chromosomes together form a chromatin network, and their individuality is now lost. The group of the chromatin network at each pole are surrounded by the nuclear membrane, and nucleolus is also present. There are two fully formed nuclei, one at each pole, and spindle fibers are absent during this stage. Here we have a diagrammatic representation of different stages of metaphase. Here we can see the interphase where the cell is metabolically active, the prophase where the condensation begins, and you can see clear thread-like thick chromosome structures during late prophase. During metaphase, the chromosomes are arranged along the equatorial plate. During early anaphase, you can see the separation of the chromosomes, and you can see the typical appearance during late anaphase. At telophase, you can see formation of the two chromatin at the two poles and development of the nucleus towards the end of telophase. Precautions. Coming to the precautions, we lift the microscope with two hands, one for the base and other for the handle. The staining should be done properly. Understaining and overstaining should be avoided. Excess stain can be removed using appropriate method. And do not touch the microscope lens and use the microscope carefully. So that's it about slide preparation for mitosis. Kindly like, share, and subscribe if you like the content.